Okay, looks like it's time to start. Well, welcome all for coming here this afternoon for the second of our Rossi lecture series today. Our speaker today is Gavin Sachs. He's a professor from Cornell in the food science, is it technology or just food science? Just food science, Department of Food Science there. And Gavin is a chemist, and he's been there for 15 years now. Yeah, it's quite a while. And I met Gavin probably around 15 years ago, and I remember, anyway, I was very impressed with one of the talks he gave in Seattle I think it was an ASCV talk. So after the talk, I went out and sort of chatted him up and was able to talk him into helping me write a book about wine chemistry. And I was very lucky that he agreed because he's a way better chemist than I am. But anyway, I guess I was going to say he's done lots of different things in wine chemistry, very diverse background, and it's always fun to look at his latest paper. But today he's going to talk about doing analysis faster, cheaper, better, and I can't wait to hear. So, Gavin.
Thank you, Andy. And thank you for the invitation to speak. This is my first in-person off-Cornell campus lecture in about 3 years. Go figure. I also remember that meal, Andy, when we first met. We got burritos and sat outside somewhere. So fast, cheaper, better. When I have students who have a broad range of backgrounds, but often in chemistry, and when we talk about analyses, they say we want to develop something, I say, it better be at least two of these three, faster, better, and cheaper. Otherwise, I'm not sure why we're doing it. Let's just use the existing technology. So I'll talk about a couple of things that we've done in our lab to do things faster, better, and or cheaper, and then try to put it to use. Okay, so we do a few things in our lab. I'm going to be focusing on a couple parts of it. One is going to be challenges and opportunities with rapidly measuring trace volatiles using AIMS or ambient ionization mass spec. And the other is going to involve the origin and fate of what I refer to as reduced sulfur compounds like hydrogen sulfide. So let's get going with the first part. Okay, so this is a slide that I stole, more or less, from my colleague Terry Akery, a flavor chemist in my department. The challenge with measuring compounds at trace levels, and trace, you can bicker about it, maybe it starts around parts per million, maybe it's parts per billion, maybe it's probably somewhere in the ppm to ppb range. There's two challenges. Number one, you're trying to detect small amounts of a compound in a foodstuff or a beverage. But then the second problem is that it's not only your compound that's there. And so if you go down to the part per million, part per billion, part per trillion level, you can find thousands, even tens of thousands of different compounds using appropriate analytical tools, which means it's not just enough to be able to detect your compound, you have to also not detect the chemical noise, the interfering compounds that are present at similar microgram per kilogram or nanogram per kilogram concentrations. For most flavor active or food quality related compounds, taste compounds, color compounds, pigments, tactile compounds, this isn't too much of a challenge. A lot of these are biologically active and present at concentrations in the tens or hundreds of parts per million or higher. Sourness, for example, we measure titratable acidity in the grams per liter range. So you can use a broad range of analytical platforms to measure compounds when you're dealing with part per thousand level compounds. Toxicity, then you start to worry about things at the low part per billion range. But then odorants are one of the only things that we want to measure in food and beverage applications, or for that matter, almost any application, where we routinely want to get down to part per trillion levels. where we have compounds that are both present at and biologically important at part per trillion levels. So there's not a lot of analytical tools that will deliver that kind of performance for us. Mostly we rely on chromatography-based approaches like GC, gas chromatography, coupled to appropriately selected detectors like mass spectrometry. Many of you are going to be very familiar with these kinds of instruments and this kind of data. Every peak you see, every spike is some volatile that's been separated out from others. It may or may not have an odor. It may or may not contribute to the aroma of the product. But with a typical GCMS run on the order of 30 minutes or so, it's possible to get down to these nanogram per liter parts per trillion levels as long as we use appropriate sample preparation to get rid of some of those interfering compounds. And furthermore, we can both look for targeted compounds. We can have a few analytes of interest, but we can also do untargeted or semi-targeted analyses where we don't know exactly what we're looking for. We can do some after-the-fact analyses to look for other compounds that might be interesting. So when I first got started, I had no idea what I was supposed to be working on since I came from a non-wine background. That is not a real movie, by the way. I was told, oh, you should look at isobutyl-methoxypyrazine. There's a lot of interest among growers, both here in New York State and elsewhere, in this compound. So IBMP, smell of the green pepper, sensory threshold, depending on whose paper you read or believe, somewhere in the ballpark of 10 nanograms per liter, 10 parts per trillion. And it's found in grapes and wines of all types, but particularly in those wines and wine grapes associated with Bordeaux varieties, that is Cabernet Sauvignon and Merlot, Sauvignon Blanc, where it can be undetectable, below a part per trillion or so, all the way up to somewhere in the ballpark of 50 parts per trillion, nanograms per liter at harvest. And there was interest in industry because there were studies showing, number one, that it could impart a vegetal aroma, and that concentrations of this compound in red grapes could predict quality scores by consumers. And in whites, it was a little more ambiguous. Sometimes consumers liked more green, sometimes they liked less green, but it was a differentiating characteristic and therefore appropriate for a targeted analysis. And then the gold standard GCMS methods available, ones that we looked at but also tried to develop ourselves, were typically taking 15, 20, maybe 30 minutes for an analysis. Okay, so that's where I got my start, trying to measure IBMP, and then usually at the same time trying to measure a bunch of other compounds as well because, well, why not? They're all there in the chromatogram. So we did collaborations with viticulturists where we looked at the effect of cluster exposure on IBMP accumulation and degradation. Short story, cluster exposure decreases IBMP accumulation. It doesn't have much of an effect on the rate of IBMP degradation. We did work showing that IBMP, it's present in the grape juice from the beginning. You can add a non-polar sorbent like food-grade silicone and strip out IBMP, get rid of the silicone, ferment, and the resulting wine has much, much lower levels of MPs, but you don't affect other wine volatiles. And we had some questions about the effects of hedging. What happens if we leave our hedge clippings in the vineyard, in the Finger Lakes, Long Island? We get a lot of rain, and people might be hedging 5, 6, 7 times a season in some extremes. They're pulling out the bird netting to do hedging. That's pretty crazy. The question was, can we just leave the clippings there? Maybe there's some sort of like signaling pathway in the plants involving C6 pathways. Short answer for a lot of data was no real effect. Okay, so those studies were fine. And I thought, okay, we've got the right tools for doing this. And then things got harder. Or at least the questions people were asking me got harder. I started working with grape breeders. And grape breeders would say, I've got hundreds of samples, and I'd like to do multiple time points. Can you measure IBMP and these other volatiles for me? Or we'd have questions from West Coast producers. I know you here will be very familiar with this. We've got lots and lots of smoke-tainted grapes, or we think they're smoke-tainted. Can you measure these phenolic compounds and let us know if indeed they've got smoke taint? Or people working in digital agriculture, we're trying to calibrate our models to measure these kinds of compounds. Can you measure this, 10,000 samples or so? Do you have the time? So ignoring the sample prep side, simply from GCMS utilization side, 30 minutes analysis, just to do 1,000 analyses, that's several weeks. And that's assuming everything goes well. And everything isn't going to go well because it's GCMS. Something is going to break in the meantime, and some service will be needed. So thinking about this problem from another direction, in situations where we're doing target analysis, where we've got certain targets we know we're interested in data, and we're kind of interested in other compounds, but not very, we have a lot of time where we're not capturing information about our compounds of interest, and very little time capturing information about our compounds that we care about. If it costs us nothing to measure that dead time, then sure, why not? But it does cost us something. It costs us time. So in situations where we want to do a routine targeted analysis, we know what we care about. We want to do it a lot of samples quickly. For example, if we're trying to triage grapes because we think they're going to be more vegetal or produce more vegetal wines, or volatile phenols in grapes as a marker of smoke taint, or in wine, or Brettanomyces derived volatile phenols like 4-ethyl phenol and 4-ethyl glycol, TCA as a marker of cork taint. Sure, we'd love to get all the compounds, but we really need those compounds. How do we get those faster, that's still good enough? So the slow step here is the chromatography step. And there's been some work on trying to do fast GC and using shorter and narrow bore, high-speed chromatography, but often that still results in 5-minute, 10-minute runs. So the question is, what if we get rid of that GC step, that slow step altogether? Can we get good enough results if we introduce our compounds following an extraction directly into the mass spec? So the tool we looked to try to answer this question was an alphabet soup of what are called ambient ionization techniques. The general idea of AI is you have your mass spec, and instead of coupling it to something like chromatography or having an inlet that's under vacuum, you have the inlet open to the atmosphere, and you can directly place a sample in front of it. And so DART is one of several ambient ionization techniques. We selected it because it's particularly compatible with volatiles and semipolar compounds, or slightly nonpolar compounds, which describes an awful lot of key odorants. So we have the DART source, which generates metastable helium, which can then ionize other compounds in the atmosphere, like water and oxygen and nitrogen, which in turn are going to ionize analytes in your sample, which in turn are sampled into your mass spec inlet. And so what you see up here, this is something from one of the company that produces DART, IonSense, and they're swabbing a tomato and they put that swab then in front of the mass spec inlet, and they claim, and I believe them, that they can get down to part per billion detection limits for pesticides, but they're cheating. And the reason they're cheating is all of their analyte is right there at the surface. Their extraction has already been performed for them. They don't have to worry about this analyte being present in the bulk of the tomato. It's absorbed to the surface. A swab is appropriate for that. This isn't going to work for compounds that are produced inside the grape cells. And just swiping a grape with a Q-tip is not going to remove IBMP from the grape cells. So we need to do something else to get that out, which means we need to macerate the sample into terms of juice or a puree. And then what? So here's some bad ideas for how to put samples into a DART MS if you're trying to measure DART trace volatiles. And we've tried them both, so I know they're bad. You can use these commercially available meshes and put a little spot of your juice or macerate right on them. And then these cards, stainless steel mesh, plop in front of your DART source. This works really well if you're a synthetic chemist and you've got high concentrations of an intermediate to synthesizing a drug. So that's what this system often gets used for. Synthetic bench chemists, they want to make sure to make the right compound. Got my TLC spot, scrape it off, dissolve it up, spot it on. Works beautifully. For volatiles, your volatile evaporates. Or it reacts with the stainless steel, depending on the compound. It's also not going to be selective for volatiles. Everything in that sample, including your sugars and your acids, are going to show up as well, and they're probably going to mask your compound. Okay. Other approach, SPME-DART. Solid phase microextraction. That's a glass capillary coated with some kind of sorbent, like silicone. This sort of works. You could extract your volatiles. The problem is if you look at the geometry of SPME, you have this narrow fiber, and you can try to position this narrow fiber in front of this gas stream. The signal quality is quite poor. The signal is low. This is really hard to automate. There are some people who've developed holders for SPME Dart, but I don't know how to do high throughput with that. Okay, so we went a different direction. So we started with these stainless steel mesh cards, and I had a student named Julian Ostremski who took these and coated them with different coatings, including here, this is a picture of coating a mesh with, I think it's a 20 micron thick coating of PDMS, common SPME coating. And then we could take that and little arts and crafts, kind of position it into the headspace of a vial containing grape macerate, do an extraction, and then put that in front of the dart source. And you pulse on the dart source, and within 20 seconds, you see here, this is probably a 100 nanogram per liter sample. You get a nice signal for IBMP, very nice. And if we vary the coatings that we use, for example, using DVB, PDMS, divinyl benzene PDMS, again, another common SPME coating, we can improve signal for a broader range of analytes. We can then do a little validation, comparing spiked samples of macerate against for GCMS, IBMP by GC, versus DART. Okay, so there's a couple problems with this. Number one is the early approach wasn't all that great, all that sensitive, which is why we're operating in the hundreds of nanograms per liter, not the tens or single nanogram per liter range. The other problem was this is still not easy to automate. This is one card, you push down, grad student. If you want, the automation is the grad student drinks a lot of coffee is the automation. So you do that over and over and over, one at a time. There's no time savings here. Yes, the measurement in the mass spec is fast, but all the other steps, including the extraction, are slow. So we had another step in the process, which was transforming this one at a time, one-shot mesh into a sheet. So this here is a sheet of silicone that's been laser etched in our microfabrication facility to make a lattice, and that allows, when you put this in front of the dart, allows the gases to travel through. And we can take that SPME-mesh sheet, this etched PDMS, and put it into a holder and lay it over a sample. For example, a clip art clover. And put it over a little leaf, and don't contact it, just kind of hover in the headspace, and it will extract a volatile image. Volatiles will come up over the sample and deposit onto the SPME-mesh. We have to be careful. This only works with flat planar substances because otherwise it'll make contact with the surface directly. And so you can do this, and you can write a little message in invisible IBMP ink. That's the template. Message is hi. That's where we start. Take this invisible message that we've written on the polyethylene film and extract it out with SPME-mesh. Take that SPME-mesh sheet, bring it over to a dart, and read out the message. And the resolution is nothing amazing. The dart source gets down to a few millimeters resolution, but it's still good enough where we can see what we wrote. But that's nice, I think. We've thought about applications. Maybe you could use this for, if you've got packaging and you think you've got scalping or tainting, you could use this to localize the source of that scalping or tainting. You could, plant pathology world, if you want to know where volatiles are being produced in a leaf. Yeah, maybe that could be interesting to look at that distribution. But the way we wanted to use it was to improve our throughput for volatile analysis. So imagine the same idea, SPME-mesh sheet, put it into an appropriate holder, place it over a multi-well plate that has been filled up with multiple samples of different types. And then you extract a volatile image. That volatile image is the image of all those different samples that are spatially resolved from each other. And then you can transport that over to the Dart MS for rapid analysis. And hey, let's see if this works. It worked before, will it work again? So this was work by Madeline B. One of the entertaining things about doing YouTube is you get to see what people were recently watching here, or at least what's trending. So I have a good idea of what people care about at Davis based on what's suggested. Okay, so, or at least your previous speakers. Okay, so first step is baking out the SPME-mesh sheet. And then in the meantime, that is, I believe, fast forward, I hope. That is the Madeline filling up her multi-well plate, 24-well plate with, I think, just water or a standard solution of some kind. Okay, covering up with some spacers, and then sealing in place the SPME-mesh sheet, which is a little piece of silicone, and goes into our version of an incubator, which is a GC oven. For those who haven't tried, you can make an amazing pizza in one of these things. Like, I mean, basically, there's convection ovens, right? Do not do that unless you're getting rid of the GC oven. Don't do that in the lab, but take it out of the lab if you're going to do it. Okay, so then we incubate this. We have now extracted all of our samples, or the volatiles, onto our SPME-mesh sheet. We will carefully take out that holder containing our SPME-mesh sheet and place it in front of the dart. And now the dart source will start to go across, run, data collection. So the signal that you get from the dart, we call them chronograms, they're not chromatograms because there's no separation. The signal you get is just the dart source going across the different wells of the plate. And it takes, I think, 24 samples in 17 minutes. That could be faster because you just jump from sample to sample. We're working with IonSense right now to rewrite the driver to allow us to do that. So that's it. And then you, in the meantime, you've got your other SPME-mesh sheet baking out in the oven and incubating, and you switch over to that one, and you go back and forth, and you can run lots and lots of samples in less than one sample per minute. Okay, play from current slide. Good. Okay. So here's what the data looks like. When you see a blank, this is a blank sample, and there's a transition where it's going over the stainless steel, and then it picks up a well that's got a couple hundred nanograms per liter, then a blank, blank. So each of these corresponds to a well in the multi-well plate. The data comes out as one big long strip, like a strip chart recorder, and then we have to cut and paste to make it look like a picture like this. So good reproducibility, good RSDs. Spikes at this level, at this time, were still relatively high. We had to improve some things with the SPME-mesh holder, so we had less leaks and cross-contamination. But then there's the moment of truth, which is it valid? Can we use this approach? Less than 1 minute per sample, but who cares if it's not giving us the same data as we would get from the gold standard, which is SPME-GCMS. So we partnered with E&J Gallo because, well, they've got interest and lots of vineyards with different levels of IBMP. We also looked at linalool, marker for muscat, floral type aromas, vineyards throughout California. We had hoped to do a few years. 2020, of course, was a challenging year to get experiments going. But we have a couple years of data now for IBMP, and I'll show you one of those years. Yep, so here's what it looks like. This is 300 Cab Sauv and Merlot grape vineyards operating at range you expect to see for IBMP. Undetectable, 1 nanogram per kilogram or so, up to about 20 nanograms per kilogram, 20 parts per trillion. SPME-GCMS versus SPME-mesh DART-MS. Yeah, so one of the questions could be, so how accurate is it? Well, we're getting root mean squared errors of between 1 and 2 parts per trillion. Another is, would you make the wrong decision? Would you misclassify something? And would you accidentally take a sample that you thought was 0 to 5 and it's actually 20? If we think a sample is above 15, it doesn't have less than 5 and vice versa. So this feels pretty good. Seems to work well. We get similar performance to SPME-GCMS, the gold standard. Detection limits down around 1 parts per trillion. Respectable linear range up to what we expect to see in harvested grapes. Good recovery, good reproducibility, replicability. Big difference is one instrument, everything goes perfectly. We could do over an order of magnitude more analyses per day. This doesn't mean that you will do this because all we've done now is move the bottleneck. The problem we get to when we get to 2,000 analyses per day isn't the SPME-mesh DART-MS, it's how quickly can you mush up your grapes and add it or uncork your wines and add it into the multi-well plate. So something else then, analytical chemistry is always a matter of moving the bottleneck from one place to another. So that's the next step, is figuring out how to get around that bottleneck of sample preparation. So we've done other work. We've done work with linalool. Correlation's okay. The slope is 0.5. This means that we're getting higher values for SPME-mesh than for SPME, GCMS. This is probably because if you've done GCMS on monoterpenes, you know they all have basically the same mass spectrum. We're probably picking up interferences from things like terpineol, geraniol, other monoterpenes, but they all correlate pretty well. Aldehydes, trying to measure aldehydes in native form is not easy. In fact, it's basically impossible. But if you take the trick from SPME GCMS playbook, you derivatize them first, convert them to their oxime derivatives, then they light up very nicely, and you can get respectable detection thresholds. Something we're continuing to work on, and of interest to this group, volatile phenols. So this would be both as markers of smoke taint and markers of Brettanomyces contamination in finished wines. But probably the real interesting one is smoke taint. We had people calling us on the East Coast to do analyses for West Coast fruit. That does not happen in a typical year. It happened in 2020 because everybody got overwhelmed by the demand for analyses. And that's tricky when you've got a system where most of the time you don't need analysis, but then all of a sudden everyone does. How do you have that capacity? So the challenge with doing headspace SPME-mesh is the phenols aren't terribly volatile. They don't extract all that well into silicone. What we started doing is immersion. You just flip over the device. We had to make a few modifications to make it watertight. This works. You still, in less than a minute per sample, can get down to microgram per liter detection limits for phenols in juice. You get good accuracy for the Brett phenols, 4EP and 4EG. The problem is for the volatile phenols we care about for smoke taint right now. We're seeing interferences for some but not all samples. We generally never see too low a value, but we do get false positives, especially for 4mg. So what we've assessed right now, what I've used it for right now, would be triaging. If you get a high number, then it's worthy of looking at with a SPME GCMS, a gold standard technique. If you have a low number, we have yet to find a sample where we get a low number that wasn't also low by SPME GCMS. There's probably some additional ways that we can improve this. We're looking into using alternative SPME-mesh materials and derivatization. Again, we need to get some additional selectivity beyond what we have right now. Okay, last part of this rapid volatile analysis. What occurred to us as we were developing these materials is we weren't just doing something for rapid analysis. We were developing a way to really conveniently and stably extract analytes. In this case, volatiles, but we've got some other tools that we can do for extracting non-volatiles as well. If you've done something like this, like 23andMe, you were not forced to go to San Francisco headquarters and show up in person and spit into a vial. They have tubes you can spit into, and you collect your saliva, and it's stabilized, and then they can do the DNA analyses later. You don't transport your entire person to San Francisco. But that's kind of what we do right now for a lot of plant analyses. A person at a remote site, maybe it's a breeder, maybe it's a field site, they collect a bunch of fruit, and then they pack it up to send to a facility that's capable of doing the phenotyping. And it may be local, but maybe it's on the other side of the country. And how do you get perishable plant samples to the other side of the country? You probably do something to stabilize, like freeze it or dry it. That is a pain, that's expensive, and it seems like 10% of the time the samples show up half thawed anyway, and you lose them. So what if instead there was some easy, not too expensive way to conveniently extract samples with whatever you're interested in, volatile, non-volatile, little sheet, roll into a tube, send it off into the mail, and it shows up the same way that we would receive our DNA samples. So we do something like this for, I don't do it, but doctors do something like this for blood spot analysis for newborn infants. So do a little finger prick onto a card. And it seems like there's some good opportunities to start thinking about doing things like this for trace analytes in plant samples too. Okay, next part. Reduce sulfur compounds with a particular focus on H2S. I'm so sorry, Pat. I won't be talking very much about SO2. I figured they hear about plenty of it from you and from Andy anyway. So I'll talk a little bit more about H2S. Okay, hydrogen sulfide. Such an easy compound to have to draw by ChemDraw. Yeah, so there's H2S. Smell of rotten egg and flatulence. And yes, there's a very, very entertaining paper that identifies this as one of the key aroma compounds in flatulence. It's worth reading just for the method section. Detection threshold, low parts per billion. So the reason this is of interest, number one, is that it has an off aroma, but when you have wines that are described as reduced or sulfurous, having sulfur-like off aromas, almost all of the time they have hydrogen sulfide present at superthreshold concentrations. And we don't see that with non-faulty wines. Furthermore, at an international wine competition, or international wine competitions overseen by Jamie Good, SLOs, sulfurous off aromas, account for somewhere between a quarter up to a third of faults observed in wines. That does not mean that a quarter of wines were faulty, but of the wines that were faulty, sulfur-like off aromas were responsible for a quarter to a third of them. H2S may be directly responsible for these off aromas in some cases. It may be reacting with other compounds to make other off aromas and basically just serves as a marker then, or it may just be a marker for reductive conditions. But when H2S is around, that is a sign of a wine that is likely going to be described as reduced and vice versa. Okay, sources during fermentation have been well studied, including here at Davis. Linda Bassan, among others, has done some excellent work, or did excellent work on yeast amino metabolism and its role in generating H2S, both through de novo production of cysteine and other amino acids, and also a little bit of work on catabolism of amino acids, and particularly in low nutrient musts, such as those deficient in yan. Another well-established source, degradation of elemental sulfur pesticide residues. One of the most widely used, at least by mass, the most widely used fungicide for grape production. Yeast have the ability to convert elemental sulfur to H2S. And this reaction has been known, the earliest paper I found dates back to 1869. It's been known for a while. If you take elemental sulfur and you add it to an ongoing fermentation, it will start to reek of rotten eggs very quickly. And so there's a couple of pathways proposed. One is that this is non-enzymatic, just glutathione or other thiols. Yeast produce an extraordinary amount of this tripeptide, glutathione. You can buy these yeast extracts that brag about being high in glutathione. Glutathione will just non-enzymatically reduce elemental sulfur, make H2S. There's also been some hypotheses that because yeast appear to have thioreductases, perhaps this is enzymatic as well. The real problem is not just that it happens, but when it happens. There's Terry Aker again. Wow. Elemental sulfur, if it's present, you get production not just early on in fermentation, but towards the end of fermentation. That is, you get it when you're no longer actively producing CO2, which means it's going to have a longer residence time, which means it may persist even after fermentation is done, as opposed to this early formed H2S where it's a nuisance. It's a signal something's wrong with the fermentation, but most of it's going to get sparged out of the fermentation by CO2. Okay, so going back to when I first started, I have no idea what I'm supposed to work on. So I went to Wayne Wilcox, plant pathology, and asked him for help on ideas. And he said, you need to tell me easy ways to measure elemental sulfur, because I want to know, I want my growers I work with to know how late can I spray elemental sulfur. Number one question I get. So I showed Wayne some literature data, including from Doug Gubler here at UC Davis. He says, that's one study using dusting sulfur in California. I want to know here, because we've got different formulations we use, we apply at different rates, our weather is quite different. We get rain here during the summer. They use a different sprayer than we use, and people don't always do the same. I want to have a method where we can use it and do experiments, and I can hand it off to growers, and they can make their own measurements. So that wasn't available. There were some methods, they were challenging, mostly HPLC based. So we wanted to have an easy, cheap test that we could hand off to winemakers, grape growers to measure elemental sulfur residues. And for inspiration, we looked at the way H2S is measured. Gold standard methods, Andy, you have this equipment, GC, sulfur chemiluminescence detector, or other coupled techniques, GC combined with other selective detectors. The challenge is that, first off, this is specialized equipment. Second, H2S is present at low concentrations, trace levels, parts per billion. It is highly volatile. It's super easy to oxidize. It readily reacts with any active sites that you might have, anything electrophilic, any transition metals. It's challenging to send a sample out for analysis for H2S and not have it come back as 0 because you lost it all during shipping. So Sung Park, who came through Davis to do his PhD, and I think has been a visiting scientist since, the clever idea of using these gas detection tubes. These are developed for environmental studies, used by the mining industry to detect and quantify H2S in the air. And so the idea is that these are strips that are impregnated with a metal salt that will react with H2S, and then the length of the stain, how much darkens, will be proportional to how much H2S was exposed to the stick. And so what kind of metal salts? Something that will react with H2S, lead salts, mercury salts, cadmium salts. They're usually not very nice salts. I think some of them are zinc-based. But that would work, at least it works for H2S, and we can make it work for elemental sulfur too, by converting that elemental sulfur into H2S. So we tried a number of different reducing agents. The one that performed best, 100% recovery, was DTT, which is dithiothiotol, widely used among biochemists to reduce sulfur-sulfur bonds. You can take a little flask, top it off with a volumetric as a pipette, as a condenser basically, hook that up to a sulfur stick or gas detection tube, add your reducing agent, add a little Alka-Seltzer tablet to buffer the solution of all of CO2, and you have a test that takes about 30 minutes to run, requires about $5 of consumables per analysis, no fancy glassware, gets good recovery, and has detection limits well below what's been suggested to result in problems during winemaking. Okay, so now we can take this test, put it to use. So we did some trials with different spray rates, timings, Chardonnays here with different amounts of sulfur residues, and what we find is, as previous literature shown, if you've got around 1 ppm, 1 milligram per kilogram elemental sulfur at harvest, you're going to have elevated H2S production during fermentation. Furthermore, we could do studies looking at different pre-harvest intervals, different timings of the last sulfur application. We had growers in New York State who were being told by winemakers, do not spray elemental sulfur after fruit set. That the idea that somehow elemental sulfur was going to survive 100 days, if that was the case, we'd only have to spray once in New York State, and we do not have to spray just once in New York State. I don't know why they were putting those limitations. What we could do is we could say, here, look, we don't see any evidence that sulfur residues are persisting past 7 weeks. In fact, in a number of these applications, they seem to be gone in 5 weeks, but here's the test. Test for yourself. Don't let yourself get blamed for a bad fermentation when it had nothing to do with the way you were managing your grapes. So in most cases, when we've done follow-ups with growers, winemakers that have had the forethought to freeze or hold back sample or to test ahead of time, we don't see much evidence of elemental sulfur where we are as causing problems. We've got other things going wrong with fermentations. So that's how we got started. Where this went next was questions from winemakers of why are wines we're putting into bottle going in fine and coming out stinky, reeking of hydrogen sulfide, having these sulfur-like off aromas. And this corresponded with winemakers finally improving their bottling lines enough such they were no longer oxidizing their wines. This was a disease of affluence. They finally invested in decent bottling equipment. And so this has been reported in the literature, although not too much until recently. You can take the same wine, put it under synthetic closure with lots of decent amount of air in, versus a sealed ampule, glass ampule, which lets almost no air in, or should be no air, should be a hermetic seal. And you can see this continuum of H2S following storage. And this wine originally had no detectable H2S. And so this wasn't the only study to see this, that if you store wines under anoxic conditions, you can find H2S forming as well as other sulfur-like off-aroma-causing compounds during storage. So that's interesting. The main focus of this often was what's the right closure, but what I was interested in is, so where is this H2S coming from? What's the latent precursor of this H2S? Okay, so I'm going to show work that's not my own. This is Gal Kriegman's work. He worked with Ryan Elias at Penn State. What happens to H2S and related sulfhydryls when you oxidize a wine? So before we talk about reappearing H2S, let's talk about where it goes first. So there's a couple of pathways. One Andy has worked a lot on, the iron phenolic pathway. If you oxidize a wine, phenolics can get oxidized to corresponding quinones. Those can react with thiols and, or I should say, sulfhydryls more generally, including H2S. Another pathway, which Gal worked out, is that oxygen can react with transition metals, particularly copper, but also iron 2, and generate a corresponding oxidized form. And this can then react with hydrogen sulfide and thiols to generate we'll talk more about these, copper sulfhydryl complexes, which stay in solution, and what historically we just would have probably called disulfides, but more properly seem to be a mixture of mixed disulfides and polysulfanes. That is, they're not necessarily symmetric, and sometimes they've got multiple sulfurs in the middle. Okay, so that seems to be the main source of H2S precursors, is these oxidation products here, because, actually, before, one other point, this copper 2, you can form it two ways. Number one is you can oxidize existing copper 1 in solution. The winemaker can also add it in the form of copper fining. This is the reaction that seems to be happening. You end up with both formation of these complexes and formation of these mixed disulfides and polysulfanes. Okay, and then what? So you do this copper treatment, or you do oxygen addition, and you make these precursors, and then in the presence of bisulfite and maybe some other reducing component in wine, you reverse the reaction. And so polysulfanes can react with bisulfite to release some of the original sulfhydryls, including H2S. It's not entirely clear still what happens to those copper sulfhydryl complexes, but during long-term storage, they are under reducing conditions, they are not stable. It seems plausible that they react with bisulfite as well to reform the original thiol and H2S. Okay, so this seems to be, at least I can tell, at least two of the main pools of latent H2S and other thiols in finished wine. It's oxidation products of those thiols and H2S from further back in the wine's history. Okay, so we wanted to study this for elemental sulfur. And before we did that, we wanted a protocol for measuring H2S that was easy to do and cheap, because we wanted to do lots and lots of samples. So we did one that was pretty similar to our previous method, only we left out the DTT. I think, Pat, you were the one who suggested using a condiment bottle because it's really easy to seal off. Thanks for that suggestion. It does work. Another approach, and we like this one a little bit better, is you can modify an AO unit or any kind of glassware you have around and do basically an old school Monier-Williams type sparging and sparge out your H2S into a gas detection tube. We like this because antacid tablets change the pH, and one of the concerns we had is that we were accidentally releasing some of our precursors when we didn't want to. Here we can operate at ambient pH, wine pH. Okay, so to measure free H2S in a wine, you just use the wine as is, start sparging. If you want to measure precursors, you can add brine. Vicente Ferrera's group has shown that these will release H2S and other thiols from metal sulfhydryl complexes, probably primarily copper sulfhydryl complexes, or you can add a reducing agent like TCEP, which will not touch brine releasable H2S, at least not very much, but it will act on polysulfanes to release H2S. And with this method, we can get down to sub-part per billion detection limits for an appropriately sized wine sample. Okay, so what happens to these sulfur residues? We do fermentation. What happens then? And so we had done an experiment. The starting point was we had done an experiment. We were looking at what happens long-term after we did these experiments with grapes spiked with different levels of elemental sulfur preparation. And we took those wines. By the way, if you're wondering, were those wines bad? Oh yeah, they were really bad. Okay, so we spiked them with elemental sulfur. They were gross. Then you take inert gas and you sparge out all of the H2S, and they smell fine. No more detectable H2S. And then you store those wines under an appropriate closure and store them for 3 months. And at bottling, there's no H2S, but after a few months, H2S is creeping up in those wines that were fermented in the presence of elemental sulfur. So what's this precursor that's forming elemental sulfur? We did not think that it was elemental sulfur for a couple of reasons. The big one is elemental sulfur is not in any way soluble in wine. It's highly nonpolar. And we also done some work fractioning with HPLC showing that we could separate some with HPLC. It was relatively early eluting. It was pretty polar. It was an elemental sulfur. Okay, so it wasn't. So we could take these same wines that produce H2S in a few months, and we could treat them before bottling with TCEP, that polysulfane reducing agent, and we can release H2S. If we do the same thing with brine releasable H2S, that is adding brine. We don't see any release of H2S. So what we think is going on is that these wines that are fermented in the presence of elemental sulfur have these latent precursors in the form of these polysulfanes that we can then release with TCP. And what is that latent something? We think it's this. We think it's the reaction product of thiols like glutathione forming polysulfane intermediates, some which will continue all the way to H2S during fermentation, but some which stay as glutathionated or other related polysulfanes, which will then slowly release H2S during storage, and we can follow up with HPLCMS, and indeed there is are these polysulfanes, at least this trisulfide, present in wines that are fermented in the presence of elemental sulfur. Okay. Let's see, so we can add elemental sulfur to our list of things that you can do to a wine, intentionally or unintentionally, to create these H2S precursors. I want to talk about this? Yeah, this is, I think I'm going to skip this because it's sensitive to time, but it looks like glutathione also has a role in stabilizing copper sulfhydryl precursors of H2S in wine as well. So the last application or the most current application that we're using our H2S approach for is this, wine and aluminum cans, which is the fastest growing sector of wine packaging. Very easy to be the fastest growing sector of wine packaging. We start from basically nothing. It's still a small percentage of the market, I think under 1%, but that's up from 0.01% only about a decade ago. So reasons for the popularity. I asked my students, I have all these reasons up here, like, so why do you drink wine from cans? And the answer is, yeah, because I can go to parties and drink from the can when everyone else is having a beer. Seems to be the most common reason, or I can go to the pool and bring it. But there's a number of advantages. Aluminum's recyclable, and there's good recycling rates. So from a sustainability standpoint, both recycling and also shipping, a lot less weight to move around. You're not moving around heavy glass. Okay, but there's a problem. And so this is a convenient sample of bottled and canned wines. You can't find perfect matches, but we tried the best we can to get canned wines from local stores and then find same vintage under a good quality closure, same wine style and type. And what you see is H2S is routinely above sensory threshold, well above sensory threshold in canned wines. And if you go to bottled wines, well, if you look where the median is, it's down here. I think the most common value we get for bottled wines is undetectable for H2S. Occasionally, we find an errant wine that's slightly above. So what's going on with canned wines? And so let's go look at the literature, and there is no literature. What there is patent literature and lawsuits to look at. So here's the patent literature. You can find patents involving things that will affect the impact of free SO2 on sulfidic characters, and they'll put in equations like that. Aluminum plus SO2 forms H2S, and then we have this lawsuit here by a cider producer claiming that the cans that they were sold created a foul odor. If you read carefully, it involves rotten egg smells. Okay, so how is this happening? What's happening? Is this even real? So the idea that SO2 can react with aluminum, or wine can react with aluminum to generate H2S has been known for some time. So if you look carefully at the authors on this 1937 paper right there in the middle, Lyman Cash of Cash Still fame, this was his greatest work, in my opinion. They went and got a whole bunch of different metals and dumped wine on it, or other way around. They took metal shavings and added to wine, and then wrote down what they observed. And so if you do that with aluminum shavings, you make H2S. This is a really interesting paper, by the way. I strongly recommend reading it. This is right after Prohibition, so no one had any idea what they were supposed to use. And you can do this in the lab too. Here's aluminum foil. You stick it into wine, any wine, and you'll make H2S. As long as that wine's got SO2, you can do the same thing just with simple SO2 solutions. You'll see these blisters form. You'll get H2S forming. Okay. So how is this possible in a can? Because cans, I tell my students, an aluminum can is not actually an aluminum can. It's a plastic bag. The can's just there for mechanical support, to keep the bag from splitting open. The inside of a can, you can see it here, it's this thin plastic coating, thin polymeric liner, and it's supposed to protect the can from the beverage. That's what keeps it from corroding, or at least it's supposed to. And these liners historically were based off of BPA epoxy. That's been or being phased out due to health concerns. There's, I think it's Prop 65 in California requires labeling potential health hazards associated with BPA. So now you see producers switching over more to BPA-based non-intent epoxies or acrylics or polyesters, polyolefins. Why is that not doing its job? So we've been looking at this from a few different directions. One is we've been trying to develop accelerated aging assays. Can we predict in a couple days or a week or two what's going to happen in 6 months? Another is components. What components are responsible for H2S formation? Is it just free SO2? Is it free SO2 at all? And then the last is, can we do something to stop it? Can we prevent corrosion from happening? So I'll talk a little bit about 1 and 2 right now. We set up experiments, sample of wines from the grocery store, suggested by collaborators, mostly whites, since that's what most canned wines are, is mostly whites, a couple of reds, a couple of rosés. And then we measured chemistry. We did aging studies out to 8 months in cans provided by an industry cooperator with different liner types. The same wines were then incubated with clippings, that is cut out coupons, little strips of the aluminum lined with those same materials, again, provided by our cooperator. Edges there are sealed off with hot melt glue to keep the bare aluminum from reacting with the wine. And what do we see? To summarize a whole lot of grad student time, best predictor of whether or not you're going to have H2S during long-term wine storage, molecular SO2. But pH and free SO2 are also pretty high. And those are really hard to uncouple from each other. So we're trying to do some follow-up studies to figure out which of those is more important, pH versus free versus molecular. Based on what we've seen so far, I think molecular is probably the culprit, or at least the best predictor. It could also be pH plus free SO2. Things that don't matter very much, total SO2, TA, alcohol, at least over the range we see in wines. Copper, the wine, Ball Corporation, other can producers are requiring people to measure copper. We don't see any correlation between copper and chloride in can failure and H2S formation. What we do see is a correlation between SO2 and particularly molecular SO2. So what we're working on now is the mechanism. How is this SO2 exerting its effect? Is it somehow diffusing through the liner? We don't think it's pores in the liner, and that's because if we use bare aluminum, we don't see a good correlation between what we get with bare aluminum and lined coupons. So it's not just simply holes in the coupon or in the liner. There's something else more subtle going on. It may be that SO2 is able to degrade the liner and then facilitate other corrosive components to get to the surface. Accelerated aging, pretty good results so far. Good correlation between what we see. Looks like around 7 days seems to be ideal at 50C, correlates nicely to what we see out to around half a year storage time. You get pretty good fit between those two plots. So we've got a nice test now for doing rapid assessments of the effect of different wine composition and liner types and also anti-corrosives, because there's plenty of industrial applications that struggle with corrosion, for example, aluminum engines in marine environments, and they are exploring the use of what they refer to as green anti-corrosives to prevent corrosion. Classic anti-corrosives involve things like arsenates and chromates, not appropriate for food use. There is no such thing as food-grade arsenate. So what you can find, though, is hydrochlorides. Carboxymethylcellulose has been tried out for an anti-corrosive. Interesting. So this is what we're doing right now. We're trying out different macromolecules, mostly hydrocolloids, a few other compounds as anti-corrosives, and evaluating their effect, not just with wine, but with a broad range of what are referred to as hard-to-hold beverages. This includes things like energy drinks, sour beers, kombucha, things that are known to cause corrosion in cans, and people have thus avoided their use or cautious about their use as a result. Good. We're at the end. So I'll put up the summary slide and acknowledge the people who did all the work, which are the students, Joe Yastrzemski, Madeline B., Jesse Rafson, and now this is not Terry Bates, the viticulturist, this is another Terry Bates, unrelated, have done all the work on rapid volatile analysis, and H2S formation was piloted by Misha Kwasniewski, and a little bit of work by Joe Yastrzemski, and then more recently, Rachel Allison and Austin Montgomery have been driving this. Thank you very much for your time and attention. Thank you again for the invitation, and I'd be happy to take any questions. Yes, ma'am.
Have you guys investigated utilizing agitation or some type of sparging mechanism within an inert gas for your volatile studies?
Yeah. So the question was, when we're doing a SPME-mesh analyses, do we agitate the samples?
Yeah, because as an alternative for using the incubator.
Yeah. So the combination of the two goes fastest. So we've done both there. That was an earlier version where we just did incubation. We now have a heated incubator, or we no longer rely on the GC oven. So the combination of the two, if you do both, just like SPME, if you do agitation plus heat, that's fastest. The only reason why, so agitation gets you there faster, so the SPME-mesh, but that's usually, what limits us now for the kinds of compounds we want to go after seems to be more the chemistry of silicone. There's a reason why there's so many choices available for sorbents, for SPME, because different sorbents have different selectivities. PDMS is really easy to work with. We've tried making larger format versions of things like divinylbenzene. It's hard. They get really brittle. So, agitation is really good, heating's good, but changing the materials, I think, is going to be the way that we get better selectivity and sensitivity.
On the flip side of speed, in terms of accuracy, when you're looking at the two techniques of doing heat and agitation, have you looked at those in isolation, and could you speak to the accuracy?
So for accuracy, this would be like with recovery spikes, for example.
Yes.
We have not. We use isotopically labeled standards. That covers up a whole lot of bad analytical chemistry when you use deuterated standards. So you can make a lot of mistakes. The main issue there, if we don't agitate or don't heat, is we don't get very much signal and our detection limits are lower. So that will hurt accuracy just because you can't see anything. But no, we haven't tried to independently suss out the difference between those two. Done.
So, coming from a biology perspective, one of our goals is to understand the cell as a system, right? And I was, I guess, surprised in part today that you're asking some of these questions, like where did this stuff come from, right? And suggesting that we don't understand the wine system, as a whole. So just thinking about that as kind of like an open-ended question you can answer any way you like. How close are we to understanding the wine as a system, and what's the future there in terms of big goals? Yeah,
Ben asked what's wrong with us chemists? Why haven't we figured out where everything comes from in wine? Because you have an easier system. We do have an easier system, right? Yeah. So, what I would say is for wine, so the question is, how well do we understand, if we would take a finished wine and we knew where it started, how well can we predict going forward or rationalize going backwards? The short answer is that for some classes of compounds, we have a pretty good grasp of what matters. Anything at relatively high concentration, that's relatively stable to measure, and especially if it undergoes like acid catalyzed transformation of some kind, ester hydrolysis, we've got a pretty good grasp of what's going on. There may be some really fine points, but right now, if you told me the concentration of isoamyl acetate in a wine right now, banana aroma, we can predict where it's going to be. So as long as we know, say, pH and the storage temperature, we're going to be pretty close. These compounds, a lot of sulfur chemistry, this isn't just in wine, was very poorly studied until the last 10, 20 years ago because it was hard to measure for all the reasons that I've just said. And so people would ignore it, not because they didn't think it was important, just because it was such a pain. And so there are still areas often involving hard to measure compounds that either have very heterogeneous distributions. Tannins come to mind, where you don't have the ability to measure just one discrete compound. It's a huge range of compounds. Or the compounds are challenging enough to measure where it's been tough historically to make a number of measurements. Or in the case of copper sulfhydryl particles, they're at this scale, hundreds of nanometers. We don't have a lot of good analytical tools for that kind of scale. Really big, we're fine. Really small, we're fine. That in between is rough. So there's still unanswered questions. And sulfur chemistry is one of those areas of unanswered questions. Eddie.
You showed the polysulfanes converted by a reduction step of some sort.
Yeah, I had shown, and this is Gal's work with Ryan, had shown that, actually maybe it was Marlise Becker's work at AWRI. If you add bisulfite, you can reduce those. You can also have thiol-disulfide exchange as well. So glutathione could potentially react as well.
So is that the only reducing agent in wine that'll break those down to H2S, or are there other things going on?
So the two obvious ones in terms of concentration and reactivity and no reactions are going to be bisulfite and other thiols. I mean, those are known to be able to reduce disulfides or polysulfates. So what other compounds could reduce polysulfanes? Do you want to propose one? Are you going to propose phenolics perchance? Yeah, I don't know. Those seem like pretty obvious ones to me in terms of their ability to reduce those classes of compounds. Yeah, so a whole lot of interesting chemistry happens in wine because we want to add SO2. It's, and this may be another example of that.
So if there are more questions right now, I'd like to encourage everybody to join us for a wine reception across the courtyard on the way out, and keep chatting with us, keep chatting with Gavin. And thank you all very much for coming.
Thank you.